All right, everybody. Thank you for being flexible and working with me here. I believe this is where we left off, excuse me, the other day. So we were talking about functions of the stomach. We left off with talking a little bit about B12 and how the stomach impacts its absorption. Remember, it does not get absorbed in the stomach, but because of intrinsic factor here, that will affect B12 absorption further down. Excuse me, in the small intestine. Okay, so we'll go from there. Um, Come on now, switch slides. There we go. All right. Now, one of the things that we need to point out about the stomach and its anatomy and how it affects physiology is something called the gastric mucosal barrier. Remember that gastric contents are highly acidic and so strong it could actually destroy the stomach itself if it wasn't for this particular feature. Now, don't worry about the cell types that produce this mucus. Just remember that there are cells in the mucosal lining of the stomach that produce this thick layer of alkaline mucus. And what it does is it coats the inside of the stomach and it resists the acidic nature of the gastric juices, and therefore it protects the stomach lining itself from being directly damaged by the stomach acid. Now, we're not talking about, in terms of, or ask yourselves the question, can anything get through it and be absorbed through the stomach lining? In general, not really. There are a few things like fat soluble compounds, aspirin, alcohol, those can be absorbed. Bacteria can sometimes actually eat away at the mucosal barrier and expose the lining of the stomach to stomach acid, which is when peptic ulcers could form. So, this type of bacteria here can actually, wear away and cause there to be a lesion or, this literal hole inside of the stomach. So things can happen. Now, if you're a doctor and you're thinking, hey, man, how do I treat this? Well, thinking about it logically, I would probably prescribe some sort of antibiotic to knock out the bacteria and then some sort of acid reducer to help protect or to help reduce the amount of acid while the stomach actually heals itself. So it's generally, I believe, a fairly easily treatable thing. Oh, I apologize for that. I thought I hit the pause button, but then I looked down and it's still recording. So my apologies. At least you guys didn't have to watch me on camera. So gastric ulcers, okay, peptic ulcers. So that's what we would typically refer to them as. So the gastric mucosal barrier is going to be very, very helpful in protecting the stomach. Okay, so just quickly a little bit about the motility and propulsion involved in the stomach. Something are these cells called the cells of Cajal. Now, the stomach normally has about 1 liter capacity for food, but has right around, I think, a 4 liter or so max capacity when it stretches to accommodate more food. Now, these cells, what are called cells of Cajal, they will generate action potentials that will cause peristaltic waves to move through the stomach, as it says there, toward the pylorus at a rate of about 3 per minute. And if you remember from your anatomy days, the distal end of the stomach is called the pylorus. And the pyloric region is that whole lower portion of the stomach, and what's called the antrum is the largest area inside of the pyloric region. And so these cells will generate these action potentials at a rate of about 3 per minute. That will then move the contents of the stomach down towards the pylorus. Remember that probably the overall function of the stomach, one of the more important ones, is to liquefy the bolus. So we swallow the bolus, it goes down, enters the stomach through the cardiac sphincter, and then mixes, and the stomach will kind of physically just pummel and beat down the food that we have there and liquefy it and turn it into chyme, chyme. I believe we talked about that on Monday. Okay, so just wanted to mention the cells of Cajal. And just real quick, talk a little bit about how quickly the stomach empties and some of the factors that affect that. So we're looking at, the stomach, let me go back up a slide here. It says the stomach can usually stay, food can usually stay in the stomach for hours after, a meal is consumed. But there are various things that can kind of speed up how fast the food is processing. But at the end of the day, one of the more important factors that affects the emptying of the stomach is how fast the duodenum and the small intestine in general are processing the chyme that gets squirted into the duodenum from the stomach. And so things like how big the meal is, the liquid nature of the meal, is it higher in fat or is it higher in protein or is it higher in carbohydrates? So the nature of the meal itself really does play a role in how fast the stomach empties. Um, Carb-rich meals and protein-rich meals actually are processed in the small intestine faster than fatty meals, and so that will cause the stomach to empty faster if the meal is higher in carbs or proteins. But if it's a fattier meal, the stomach will empty slower, and you've probably experienced that in terms of, when you have a meal higher in carbs or proteins, you don't feel as full longer than you do when you have a meal that's much higher in fat content. So that the nature of the meal itself will have some bearing on how fast the stomach actually empties. Okay, now moving on to the small intestine. We're looking at the largest, probably by overall surface area, the largest section of the gastrointestinal tract, and it's modified to have a tremendous amount of surface area for absorption. And I would always argue for the small intestine being the most important digestive organ in our bodies. Now, how do we get into the small intestine? Because remember, you have a sphincter list that you are needing to populate. So when the stomach is ready to squirt chyme into the duodenum, which is the proximal portion of the small intestine, the pyloric sphincter, which we have listed here, will open up. When the pyloric sphincter, which separates the small stomach from the small intestine, so when the pyloric sphincter opens up, the stomach will contract and it will squirt little bits of chyme at a time into the small intestine, and digestion will the digestion of the chyme will continue, and the quicker it's processed, then the quicker that the stomach can squirt more chyme into it. It's not a matter of the pyloric sphincter opening up and the stomach just giving one big contraction and squirting a bunch of chyme in at the same time. It typically doesn't work that way. Okay, so moving through the pyloric sphincter, the chyme will enter the duodenum. Now, from proximal to distal, the segments of the small intestine are the duodenum, the jejunum, and then the ileum. And then the ileum connects to the large intestine at a specific in a specific section called the cecum. And I'll show you a diagram of that a little bit later. Now, once in the small intestine, I told you guys the other day that enzymes are maximally active in specific pH ranges. Now, remember that enzymatic activity in the mouth needs a neutral pH. So lingual lipase and salivary amylase are active in the saliva because of that. In the stomach, chemical digestion and enzymatic activity in the stomach needs an acidic pH range, and so we're looking at around a 2.0. And so gastric lipase, pepsin, are active on the chemical digestion of fats and proteins in the stomach because of the acidic pH. Now, here in the small intestine, what we need is a neutral pH. So you can imagine the chyme that gets squirted into the duodenum is very acidic. And if the body is going to continue to process it, and for us to be able to absorb the nutrients, then the chyme must be neutralized. The acidic conditions of the chyme must be neutralized. And so again, we need a neutral pH in the small intestine for chemical digestion to be maximally active for the enzymes, because there's a bunch of them. There's a bunch of enzymes active in the small intestine, but again, they all need a neutral pH. So take a look at this. And I made the decision on the way home today to, I'm not going to put any essay questions on the test next week. So don't sweat that, okay? Because normally what we're looking at here is the first of the two essay options. So like I said, I just made an executive decision to make next week's exam just a pretty straightforward, multiple choice, true, false matching sort of exam. Okay, so let's take a look at this. So what we're looking at here is a sequence of events that kind of illustrate how the body will neutralize the acidic conditions of the chyme so that chemical digestion can actually occur. So when chyme gets squirted here into the duodenum, again, highly acidic. And remember, not only is it highly acidic, but if it's not neutralized, it has active pepsin and gastric lipase in it, which could potentially damage the intestinal wall, the duodenal wall. And so what happens is that acidic chyme will show up, and what it does is it activates. There are cells here in the walls of the duodenum called enteroendocrine cells that are kind of have a dual role. They are part of the endocrine system, which is where hormones and glands are involved, but it's also part of the digestive system. So these enteroendocrine cells are going to secrete two hormones called cholecystokinin, which is right here, and it's abbreviated CCK, and then another one called secretin. Okay, so that acidic chyme shows up, and in response to it, the duodenal enteroendocrine cells are going to release CCK and secretin. Now, what those two hormones do, they do different things. Number one, they both impact the pancreas. Okay, and then the CCK is, other than affecting the pancreas, the CCK is going to also impact the gallbladder and the secretin, and some of what's called bile salts are going to impact the liver. Okay, so here's how it works. So when and they're hormones, okay, secretin and CCK are hormones, and hormones enter the bloodstream and then go to target cells and cause some sort of response to happen. So they will go in the bloodstream and they target the pancreas, okay, simultaneously. So what they're going to do is CCK is going to stimulate the pancreas to secrete a juice that's got all kinds of digestive enzymes in it, which is super important. But remember, they can't do anything until the acidic chyme is neutralized. So secretin, what it does is it stimulates, I should say, the pancreas to secrete a juice that's rich in bicarbonate, HCO3, right here that you can see. Let me make that a little bit bigger. Oh, too big. Okay. So CCK and secretin. So let's look at that real quick together. So secretin, again, causing that bicarbonate-rich juice to be secreted, the bicarbonate in the juice is going to neutralize the acidic conditions of the chyme, which is what we need. So because of the secretin and the bicarbonate, a neutral pH is created, and now the enzymes that are in the juice from the pancreas, which was secreted because of CCK, now those can be maximally active and do their job in chemical digestion, starting here in the duodenum. So super important action by those two hormones. Now, simultaneously in the bloodstream, those two hormones are going to the gallbladder, where CCK will stimulate the gallbladder here. It's just a little muscular sac, and it will contract and secrete bile. And the bile goes through this duct and into the duodenum. Bile is not an enzyme, so make a mental note of that, okay? Bile is not an enzyme, but bile is crucial for our body in its digestion of fat. All right, now over here we look up at the liver. And if we look at this point over here, it says bile salts returning from enterohepatic circulation are the most powerful stimulus for bile secretion. Secretin is a minor stimulus. So secretin assists bile salts that have already been used, and the bile salts returning through the circulation to the liver are going to stimulate the liver to basically produce more bile and secrete it. And so between the production and secretion of bile from bile salts and secretin here in the liver, and the bile coming from the gallbladder, that's all going to go into the duodenum and be, like I said, crucial for the digestion of fat. Now, because of the liver, because remember that the main purpose of the liver digestively really is the production of bile. I don't think you'll find much of an argument there in most sources like our book. The main function of the gallbladder is the secretion of bile. Liver can produce it and secrete it. The gallbladder typically stores it and then secretes it when necessary. And then, like we mentioned, the function of the pancreas for digestive purposes is the secretion of bicarbonate-rich juice and enzyme-rich juice. Now, you may have learned in your anatomy days that those are the exocrine functions of the pancreas. The pancreas also has endocrine functions, which are the secretion of insulin and glucagon, our two main blood sugar regulating hormones. Okay, but all of this to say that is how the body neutralizes the acidic chyme and make chemical digestion there in the duodenum possible. All right, now let's take a closer look at, excuse me, at pancreatic juice. Pancreatic juice. Okay, so, oh, no, let me back up. Come on, there we go. All right, my computer's a little lagging tonight. All right, pancreatic juice. Now, in the pancreatic enzymes, what we're looking at is a bunch of stuff, a mixture of stuff. So we've got the pancreatic proteases, and you can imagine by the name, those are going to work on the chemical digestion of proteins. Now, remember this term we mentioned on Monday, zymogen. Pancreatic proteases are released in their inactive forms, so they are technically zymogens. There is also pancreatic amylase, which is going to be working on carbohydrates, big ones like starch. We're going to have pancreatic lipase, which is the most important enzyme for fat digestion. And then we're going to have enzymes called the pancreatic nucleases, which are going to be able to digest the nucleic acids in any of the cells that are in, proteins, carbs, or any of the things that we eat. Okay, now we're not going to talk about those guys, but you probably want to make a mental note that they are included in all of the pancreatic juice that is secreted. So we're going to focus on these three, and we're going to go in that order. So the pancreatic proteases. Let's take a look at them. So in their inactive form, so these are the zymogen forms, we have trypsinogen, chymotrypsinogen, and procarboxypeptidase. Okay, so again, those are the pancreatic proteases in their inactive or their zymogen forms. Now, in order for them to be active and digest proteins, what needs to happen is there is an enzyme, okay, in the duodenum wall, in the duodenal wall itself called enteropeptidase. Okay, and enteropeptidase is going to activate trypsinogen into trypsin. Okay, now let me show you this picture here. Okay, it's a good job of it kind of showing us what's going on. So here we go. Here's enteropeptidase. So we've got trypsinogen, chymotrypsinogen, and procarboxypeptidase that are coming from the pancreas in the pancreatic juice that is secreted because of CCK. All right, so remember, they are inactive. Now, enteropeptidase here, which is a membrane-bound enzyme in the duodenal wall, is going to activate trypsinogen into trypsin. That's what this arrow right here means, coming across. Okay? So enteropeptidase activates trypsinogen into trypsin. Now, this is where it gets a little funky. Okay? This arrow right here, okay, pointing down, is telling us that trypsin here, not enteropeptidase, unless I'm wrong. I believe our book will back me up on that. Trypsin activates chymotrypsinogen into chymotrypsin. And then this one here with this little curve in it is telling us that trypsin activates procarboxypeptidase into carboxypeptidase. And then the weird one is this little arrow, backwards arrow right here, pointing from trypsin back to trypsinogen. What that's telling us is that trypsin, activates trypsinogen into more trypsin. Kind of a mind-blowing thing. So enteropeptidase starts it all off again by activating trypsinogen into trypsin. And then trypsinogen activates all three. So trypsin actually activates more of itself by activating trypsinogen into trypsin. So it's kind of a crazy thing, right? So now, once we have trypsin, chymotrypsin, and carboxypeptidase, what those are going to do is those are going to work along with what are called aminopeptidase and dipeptidase. Those are brush border proteases. Okay, brush border cells are cells in the duodenum, part of the duodenal wall, uh, and they act as enzymes. So we have aminopeptidase and dipeptidase that are part of the duodenum. We had trypsin, chymotrypsin, and carboxypeptidase that came in their inactive forms from the pancreatic juice. And so now you have those 5 enzymes working together to digest proteins. Okay, so what we're looking at really is you've got, I'm sorry, did I miss one? Aminopeptidase, dipeptidase. No, carboxypeptidase came from the pancreas. I don't know why they list that here as a brush border enzyme. Okay, so let's look at this coming from a big picture. Okay, we saw pepsin in the stomach initiate protein breakdown. So when we eat proteins, they're first broken down into big polypeptides, and polypeptides are just long chains of amino acids. Now we add trypsin, chymotrypsin, and carboxypeptidase to the mix. Okay, so protein digestion is initiated in the stomach. Once we get into the small intestine, um, you know, pepsin is inactivated, and then these 3 guys are going to start their work and add to the mix aminopeptidase and dipeptidase, the 2 brush border proteases. And so you've got those 5 enzymes active in the small intestine, and their job is to take smaller polypeptides and break them down into single amino acids, and then a few dipeptides, which remember are 2 amino acids together, or tripeptides, which are 3 amino acids together. So when all is said and done, you've got what, 1, 2, 3, 4, 5, and 6 enzymes that are working on protein digestion. So we break all those down. Okay, and now the body needs to absorb the breakdown products for the body to use. So look over here in the pathway of absorption. Now, I don't usually spend a lot of time talking about how proteins and carbs are absorbed, so let me just mention this real quick because it's got terminology that you are already familiar with. All right, so amino acids are absorbed via co-transport with sodium. Co-transport, remember that is when a protein channel, a protein carrier will bind with or transport 2 things, at least 2 things. So sodium and then the amino acids in question. They're going to bind with the protein that on the side facing the inside of the duodenum facing the lumen, and then it's going to move them through and then be absorbed by the bloodstream. Some dipeptides and tripeptides are absorbed via co-transport with hydrogen. So again, co-transport. And then we look down here. So that's how they get into the epithelial cells, the duodenal cells, is by co-transport. And then how they leave is basically by facilitated diffusion. So again, protein channels on the opposite side of the cell will move them out where they will then enter circulation. And that's how the body will then use them. Okay, so that is the digestion and absorption of protein. Now, the digestion and absorption of carbohydrates is not much different. Okay, so when we eat carbohydrates, we're looking at big sugar molecules like glycogen and starch. We learned about glycogen in the muscle unit, right? Which was a long chain of glucose molecules. So those are example of what we call polysaccharides. The saccharide portion is referring to sugar. Poly, of course, a prefix that means many. And so those are the big sugar molecules that our body has to break down. Now, the body is going to break those big polysaccharides down into what are called oligosaccharides, which are anywhere from 3 to 8 glucose molecules linked together. But then ultimately, they're going to be broken down into what are called disaccharides, which are 2 sugar molecules linked together. We have 3 of those. We have what's called sucrose, which is a table sugar. We have lactose, which is a milk sugar. And then we have maltose, which is a grain sugar. And then those are going to have to be broken down further. So whether it's a polysaccharide being broken down into an oligosaccharide, an oligo broken down into a di, it really doesn't matter. Ultimately, all sugars in our body are going to have to be broken down to monosaccharides. And we have 3 of them: glucose, fructose, and galactose. And until they're broken down to one of those 3 forms, the body won't absorb them. So let's look at how this is all going to work. So pancreatic amylase, okay, is a powerful enzyme that's going to break down starch and glycogen into oligos and dyes. Okay, so it's going to do a lot of the heavy lifting and break up those big polysaccharides into smaller chains of sugars. And then we're going to have brush border enzymes. Brush border enzymes. We're going to have 5 of them. Okay, 5 brush border enzymes that are going to break down oligos and dyes into monos. So we have dextranase. We have glucoamylase. Okay, and they're going to work on oligos of more than 3 sugars. Okay, and then we're going to have maltase, sucrase, and lactase. And those 3 enzymes are going to work on the 3 specific disaccharides. So let's look at that. Okay, so here are the 3 enzymes: lactose, maltose, and sucrose. Okay, and they are going to work on lactase, maltase, and sucrase. All right, so lactose, it's going to lactase, maltase, and sucrase are the enzymes, and they're going to work on break down lactose, maltose, and sucrose. So these are the 3 disaccharides right here. Okay, so lactase, and you need to know the disaccharides and you need to know the monosaccharides that they break down into for testing purposes. Okay, so lactase is going to break down lactose into a molecule of galactose and a molecule of glucose. Maltase, okay, is going to break down maltose into glucose and glucose. So 2 glucose molecules linked together are maltose, which is a grain sugar. And then we're going to have sucrase breaking down sucrose into fructose, which is a fruit sugar, and glucose. Okay, so again, these are the 3 dyes, and they're going to get broken down into the monos. Now, once we get the monos broken down, okay, typically what's going to happen, let's skip ahead to this, and then I'm going to come back to the slide right above it for a second. Okay, so the whole point here, remember, you can't see me because I don't have my camera turned on. But remember the other day we talked about chemical digestion being summed up with the phrase going big to small. Okay, so the whole point of carb digestion here is when we eat carbohydrates, they're going to be big, huge sugar molecules, right? So salivary amylase is going to start it all off. and not do much, but it'll start it at least. Then pancreatic amylase is going to do the heavy lifting and break down the big, huge molecules. Okay, and then we have the 5 brush border enzymes: dextrinase, glucoamylase, lactase, maltase, and sucrase. And they're going to break down a lot of the oligos, and then those 3 specifically there at the end. Let's kind of enlarge that a little bit. So these 3 right here at the end, which we can see right, the sugars here, these 3 are going to break down the dis into the individual monos. Okay, so we've got 1, 2, 2 plus 5 is 7. So we've got 7 different enzymes that are going to work on sugars. Now let's look over here at how they're absorbed. Glucose and galactose are absorbed via co-transport with sodium. Why co-transport? I actually don't know. So remember, co-transport is those monos are going to bind with a protein along with sodium, and then it's going to absorb them into the intestinal cells. For whatever reason, fructose actually goes through its own transport protein by facilitated diffusion. So it's kind of a rock star, an A-lister, an A-list monosaccharide. So glucose and galactose, they go via co-transport with sodium, and the fructose is, so cool and so special that it gets its own transport protein. So that's how they're absorbed. Okay. Now, once they're in the intestinal cells, they're going to exit the cells by simple facilitated diffusion, and then they're going to enter the bloodstream. It's pretty straightforward. But let me back up a second here and mention what's called cellulose. Cellulose is more of a plant-based sugar. And so a lot of when we eat a lot of vegetables that have cellulose in them, the body doesn't have the enzyme necessary to break that down. And so when we eat sources that are rich in cellulose, it's going to basically add a lot of fiber to the diet, to promote the movement of feces through the body. It's going to help lower blood sugar, and which is maybe a reason, and I'm sure this was just burning in your mind and you were wondering about it. But if you're a fan of corn and eating corn, we all know that when you eat corn, it typically shows up in the feces when you go poo. So, and that's because a lot of corn is made up of cellulose. Okay, and so the body doesn't break it down. It can partially digest it, but not fully digest it. So that's why it ends up in our poop. Okay, so that's the digestion and absorption of carbs and proteins. Now, the digestion and absorption of fat, I mentioned this on Monday, is a little different. It's not more complex, but it's just a little different. And normally this would be essay option number 2, but because I took out essay option number 1, I'm also going to take out essay option number 2. Okay, so again, there will be no essay question next Wednesday. All right, so let's go over the digestion and absorption of fats. So the most abundant fat in our diet is typically a triglyceride. And remember, we saw fat digestion initiated in the mouth with lingual lipase and saliva. We saw it continued in the stomach with gastric lipase. Now, in the duodenum and in the rest of the small intestine, we have pancreatic lipase. And pancreatic lipase is hands down the most important enzyme in our body for fat digestion. Okay, so here's basically how it works. And I'm going to go through and explain it, but then I'm not actually going to play the video for you, but I'm going to show you where in mastering to find a super helpful video to watch that'll do probably a better job of explaining this than I do. All right, so okay, let's see, where do I start? All right, remember, or here, no, not remember, because I haven't mentioned it yet, but when it comes to fat in our bodies, okay, fat doesn't dissolve well. It's not soluble in water. Okay, and so fat in the chyme, the fatty layer in the chyme has to be mixed, and that's where segmentation really comes into kind of a forefront, why it's important. If you like salad dressing, okay, let me use this as an illustration. because you'll kind of see this in the video. Oil and vinegar salad dressing. Like I grew up, one of my favorite salad dressings is Italian dressing, and it's probably because of my mom. But when I was growing up, my parents didn't have a whole lot of money, and they typically didn't. My mom would just typically wouldn't go to the store and buy Italian dressing. She would take, just oil. I don't know if it was olive oil, but she would take some sort of oil like that, and then she would take red wine vinegar, put them in a bottle, and then put an Italian seasoning packet in it and shake it up. And then that's what we used as Italian dressing. Now, if you ever put oil and vinegar together in a bottle, if you pour them in at the same time, let the bottle sit, and you're going to see the two separate because oil and vinegar don't mix. Oil and water don't mix. But if you shake it up, then the fatty layer in the oil will spread evenly through the the vinegar. And that's how it works in our bodies. Okay. So the fat in the chyme doesn't distribute throughout the chyme very easily. And so segmentation, that back and forth mixing, will distribute the fatty molecules through the chyme and physically break them down. So that way they don't aggregate into big fat droplets. Now, here's where pancreatic lipase will be important. So as segmentation is mixing up the chyme and distributing the fat evenly, uh, in the space there, what'll happen is pancreatic lipase will now have a lot of surface area, and it's going to break down the triglycerides into monoglycerides and fatty acids. So it breaks them down. That segmentation mixes everything up, and now those what was a big fat globule now is a bunch of smaller fat droplets. Now, again, pancreatic lipase can break them down into monoglycerides and fatty acids. Now, bile is on the scene because remember it came from the gallbladder. The bile salts, which are in the bile, okay, and then something else called lecithin, they're going to coat the breakdown products that were created by pancreatic lipase. Now, when they do that, they form a structure called a micelle. Okay, and it says here micelles are 500 times smaller than those emulsified fat droplets. Emulsion or emulsified is referring to that idea where as the fat is being mixed, it's being physically broken down from a larger globule into smaller droplets. So that's kind of what they're talking about here. Okay, so once those micelles are formed, they're going to get really close. Okay, they're not going to respect the personal space of the membrane of those intestinal cells. So they're going to get super close. Those micelles are going to get super close to the apical or the membrane that faces the lumen of the small intestine. Now, once they get there, okay, those breakdown products like fatty acids, monoglycerides, they're going to leave the micelle. And because they're fatty, they're going to move through the intestinal cells via straight diffusion, simple diffusion. Okay? Now, once inside the cell, the smooth endoplasmic reticulum, the smooth ER, is going to convert them back into triglycerides, and they're going to be coated to form a water soluble structure called a chylomicron. Water soluble meaning that it will be able to exist in our blood, in the fluid portion of the blood. So the chylomicron. Now the chylomicrons, okay, they're too big. They're going to leave the cell. They're too big to actually get into the capillaries. Okay, the capillaries that are in that mucosal layer of the small intestine. And we don't have very many fats in our diet that are small enough to do that. So what they have to do is those chylomicrons are going to have to move into lacteals, which are lymphatic capillaries. And they're going to move through the lymphatic circulation, ultimately to end back up in the bloodstream, where what's called lipoprotein lipase is going to get broken down into free fatty acids. And then the body can use those for energy production. It could store them for energy production later on. But the point is, remember that the breakdown products of fats can't get absorbed directly into our bloodstream. So they've got to enter lymphatic circulation. And ultimately make their way into circulation in general, and finally acted on by lipoprotein lipase. All right, so let's do a quick little review of the enzymes that are working on fat. So we saw fat digestion initiated in the mouth with lingual lipase. Then we had it continued in the stomach with gastric lipase. Okay. And then we had pancreatic lipase do a bulk of the work. And then finally, lipoprotein lipase. So unless my math is wrong, that's 4 enzymes that worked on the digestion of fat. Bile, remember, bile is crucial for fat digestion, but it is not an enzyme. Okay, it is not an enzyme. Just remember that. All right. And so that is the digestion and absorption of fats. Okay, let's see. All right, water absorption. A bulk of the absorption of water in our bodies is going to be done in the small intestine, and it's going to be directly related to how osmotic the chyme is. And remember, osmotic and osmosis, osmolarity, specifically osmolarity is referring to the concentration of solutes in a solution. So if our chyme is hyperosmotic, which means it's super concentrated, that's actually going to draw water to it. And so the osmolarity of the chyme is going to impact the absorption of water in the small intestine. But a bulk of the water in our GI tract is going to be absorbed into our bodies through the walls of the small intestine. The large intestine will absorb water, yes, but a bulk of water absorption is going to be done in the small intestine. A couple quick things here about motility in the small intestine. There's something called the migrating motor complex. And then, of course, segmentation. So segmentation is huge. It's huge in the small intestine. If we don't have segmentation, our body really doesn't absorb fat. Okay, so it's super important in the small intestine. And then we have something called the migrating motor complex, the MMC. Now, the MMC is a form of peristalsis, and it's kind of like a housekeeping function, if you will, of the small intestine. So what happens is there's a hormone called motilin that's going to also impact motility in the small intestine. And so this hormone, okay, it says here, after most nutrients have been absorbed, segmenting movements wane, which means they start to subside, and the hormone motilin is released. Don't worry about from where. Okay, so as it says here, motilin causes peristaltic waves to occur in the proximal duodenum every 90 to 120 minutes and sweep slowly along the intestine, moving about 2 feet before dying out. So let's use our Google Physiology translation button. So what does that mean? That once most of the chyme has been processed and most of the nutrients have been absorbed, basically what's left over needs to be disposed of. But to dispose it, we have to get it to the large intestine. So the hormone motilin will be secreted, and it causes peristaltic waves to occur in the duodenum every hour and a half to 2 hours. And what that's going to do is it's going to create short distance waves that are going to move the waste, and we can just about call it waste at that point if everything has been processed and absorbed. And those waves are going to move the processed chyme about 2 feet before they start to die out. But once those waves die out, new ones will pick up where the other ones left off, literally sweep or keep moving the processed chyme forward to the terminal portion of the ileum. Okay? Now it says here, the process repeats, sweeping the last remnants of the meal plus bacteria and sloughed off mucosal cells and other debris into the large intestine. This sloughed off mucosal cells, maybe you learned about this in anatomy, but mucosal lining of the small intestine is replaced every, I believe, 3 to 6 days, something like that. So you literally get a new intestinal lining every few days. And so as those cells are kind of sloughed and pushed off by new cells, these or this MMC, along with that processed chyme and bacteria and other debris, again, think of it as sweeping. The MMCs will sweep all of this material that needs to be disposed of towards the large intestine. So we get to that terminal portion of the ileum where it connects to the cecum, and on the ileal side, once it gets there, the pressure on the ileal side will open up the third of the 5 sphincters I told you guys about. This one is called the ileocecal sphincter or the ileocecal valve. That valve, that sphincter, will open up, and then all of this waste will then be moved into the cecum, which is the proximal portion of the large intestine. And then once on the other side, the pressure there will cause this valve or this sphincter here to close. And that's how we get from the small intestine to the large intestine. All right, so real quick, role of the large intestine to absorb some things, vitamins, electrolytes, minerals, a little bit of water. That'll get processed by the large intestine and then typically be absorbed there as well. So, temporary storage site for for waste that needs to be disposed of from the body. But kind of like it says here, it says absorb metabolites produced by resident bacteria as they ferment indigestible carbohydrates that are not absorbed. So we have good bacteria in our large intestine that will kind of metabolize some of the indigestible material that made it there, indigestible carbs. Now, this, if you were wondering, is where we or why we get gas. Okay, where if you were my son, I would say, oh, this is where farts come from. And a 14-year-old junior higher is going to find that probably hilarious. So here's a diagram of the large intestine. Okay, so here's the terminal portion of the ileum. And so those waste products are going to get here. They're going to force open the ileocecal valve, and it's going to come into the cecum here. Now the appendix here is technically part of the large intestine, but it's typically tucked back underneath the cecum here, and we don't normally see it sticking out like a finger right there. And so we go from the cecum, okay, the proximal portion here, and then we have a bulk of the rest is the colon. So we have the ascending colon, and then we have what's called the right colic flexure here, or the hepatic flexure. And it's called the hepatic flexure because of the liver being right over here. And then we go through the transverse colon over here to the left colic or splenic flexure because the spleen is over here. And then we move inferior and we go down the descending colon into the sigmoid colon. And then we jump down here into the rectum. And then finally into the anal canal. And the anus is the actual opening where feces will leave the body. Or if you have to take, if you're a parent and you're going to use a rectal thermometer on your infant, or if you have to take a rectal temp on an elderly person in a nursing home or in an ER, I had to do that a few times. You'll insert the rectal thermometer into the anus again, which is the opening here. Okay, so as you can see anatomically, okay, the large intestine is bunched up into little pockets called haustra, and haustrum is the singular form of the word haustra. And that's important because, again, we have smooth muscle lining the large intestine. And I'll tell you something about something called a haustral contraction in just a minute. Okay, so that's where things are going to be moving through until we get down here into the rectum. Once down in here, that's just about the point where it's going to get released from the body. Okay, so let me shrink that just a little bit. Let's move ahead. Okay, a couple things here. Haustral contractions. Haustral contractions are a form of segmentation, so make sure you remember that. As it says, slow segmenting movements that last about 1 minute and occur every 30 minutes or so. So these are segmenting movements that are kind of moving things back and forth, moving the waste back and forth to kind of mix that residue, mix the waste, and free up maybe some water that the body might want to reabsorb. Okay, and so kind of be thinking about water reabsorption as things move through the large intestine or small intestine and then large intestine. And like you guys know, what happens? I mean, because normally feces should be soft, semi-soft. But what happens in our bodies when the body absorbs too much water? Okay, what happens to feces when the body absorbs too much water? Okay, feces can become hard and we get what? Constipated. Okay, if the body doesn't absorb enough water, what happens? Yeah, we get the runs, we get diarrhea. Okay, um, and then of course stool is very, very liquidy, very, very runny. So, what feces look like does depend entirely on how much water the body actually absorbs. These haustral contractions are segmenting movements that are going to help free up maybe some water the body might want to reclaim. There's also something called mass movements. Mass movements are a form of peristalsis. And these movements are going to be what move the feces through the colon towards the rectum a few times per day. And generally, something called the gastroileal reflex kind of helps influence all of these movements here in the of moving feces. As it says here, stomach activity increases the force of segmentation in the ileum and relaxes the ileocecal valve. So it actually helps move things from the small intestine into the large intestine. So get things in there faster for the body to eliminate faster. And then this is the one I was thinking of, called the gastrocolic reflex. Stomach activity also activates the gastrocolic reflex, which influences the more distal parts of the colon and moves feces toward the rectum. Now, so what that's if we translate that, what that's telling us is that stomach activity, and sometimes depending on what you're talking about, certain foods and or liquids can actually increase this. And so you may notice, like let's say a strong cup of coffee in the morning that's acidic. Um, can actually influence the body to go to eliminate feces. So you may find yourself, if you have a big strong cup of coffee in the morning, it may not be long before you're sitting on the toilet, eliminating feces because of this particular reflex, gastrocolic reflex. So stomach activity influences moving feces towards the rectum. So it's kind of a cool reflex. Now, speaking of reflex, let's start to wrap things up here. Defecation. We need to talk about going poo. Now, poo, okay, so mass movements, again, remember those are peristalsis, like a mass peristalsis. They're going to move the feces into the rectum. Now, when feces move into the rectum, it activates what's called the defecation reflex. Now, it's a parasympathetic reflex. And as it says here, it's a cord level reflex. So this is all carried out at the spinal cord. Okay, so when feces move into the rectum, it's going to stretch the rectal walls, and then signals are going to go to the spinal cord. Actually, let me go to this slide here. Let me shrink it just a little bit. There we go. All right. Distension or stretch of the rectal walls due to movement of feces into the rectum stimulates stretch receptors there. Receptors transmit signals along afferent fibers to spinal cord neurons. So translation, when feces are moved down into the rectum walls are going to stretch. They're going to send signals to the spinal cord. Okay, basically that's what this is all telling us here. Okay, now part 2, a spinal reflex is initiated in which the parasympathetic motor fibers stimulate contraction of the rectal walls and relaxation of the internal anal sphincter. Okay, so let's start to put these two together now. So through the mass movements, feces are going to be moved down into the rectum. The rectal walls are going to stretch. They're going to send signals. Okay, they're going to send sensory signals to the spinal cord. Okay, which is then going to initiate motor signals to come back out, causing the rectal walls to contract and simultaneously causing the internal anal sphincter to relax. Now, I made a note here for you guys. The internal anal sphincter is smooth muscle. The external anal sphincter is skeletal muscle. Okay, so those are the last two of sphincters on the list of 5 that you guys need to know. All right, so once that happens and the rectal walls contract and the internal anal sphincter relaxes, feces are going to move down into the anal canal. Now, thankfully, the external anal sphincter is still tightly contracted, because if it didn't remain tightly contracted, we would just crap every time this reflex happened. So, number 3, if it is convenient to defecate, voluntary motor neurons are inhibited, allowing the external anal sphincter to relax so that feces may pass. All right, so if it's when we feel that, when all this is happening, is when our brain is informed about things, and that's when we perceive the need to, go to the bathroom and go poo. And like it says here, if it's convenient, that's what we do. We voluntarily relax this guy, okay, and then we defecate. We eliminate feces from the body. Now, this reflex can kind of be, let me see, how do I say this? Can kind of hit pause. So if it's not convenient for us to defecate, let's say you're driving to the Kalinga campus from Lemoore, and you get out past our college, and you get out past the base, and there, you guys know, if you've made that drive, there's nothing out there on 198. But let's say you start feeling the urge to go poo, and you're like, oh man, I better make it to that Shell station there at Harris Ranch so I can, go poo. So this reflex kicks in, and you feel the urge to go, but you can resist it. Okay, you can resist it, and that feeling will go away. But the rectal walls will be stretched again. Okay, will be stretched again, and it activates this reflex again. And so what'll happen is you'll feel the urge to go. And if it's not convenient yet, maybe you're lucky and you resist it and that urge goes away. However, that can keep going. But remember, well, not remember, I haven't told you yet, but pay in mind now that if you haven't eliminated feces and you keep resisting the action of this reflex, at some point, okay, and this is where it gets ugly, at some point your body can override your conscious control and make you eliminate feces, even if it's involuntarily, and that's the point you don't want to get to. So you better risk a speeding ticket and get to Harris Ranch safely, so that way you don't have what I call a GI emergency. Okay. Now, one more thing here in terms of the act of defecation. It's not listed in this diagram, but it is in, like I just talked about it today, actually, in my anatomy class. There's a muscle right here called the levator ani muscle. And what happens is when we voluntarily relax the external anal sphincter, and we eliminate the feces from the body, the levator ani muscle is actually going to contract. So it's on both sides of the anus. And what it's going to do is it's actually going to lift. It's actually going to lift the anus, and that's going to help push the feces out of the body. And I mean, it sounds kind of gross, but one of my students today made the good point. He's like, hey, yeah, that's Roger. That's kind of like basically what happens with a Hershey's Kiss, kind of the same the same mechanism. So it pushes it out, and the machine kind of lifts up, and that's what gives that little tip on a Hershey's Kiss. And so if you've ever wondered why sometimes poo has got a little bit of a tail on it, that's why, because those muscles contract, lifting the anus up as the feces to help expel the feces from the body. And so feces sometimes can get narrow at one end. I know a little bit too much information, but that's physiology for you. All right, so that is the defecation reflex, and that is the end of digestion part 2. All right, so that will do it for now. We are going to play Jeopardy on Monday, and we are still going to have the exam next Wednesday, so that way we don't lose any ground on the rest of the semester moving forward. If you have any questions, please feel free to reach out, and I will get back to you as soon as we can. Take care, guys.